Okay, the last part of the lecture will cover complementation analysis and then sex-linked traits. So complementation analysis. So complementation analysis, this is the question of whether or not individuals with the same phenotype have the same genetic basis. Typically used in genetic screens, so typically used in things like Drosophila or C. elegans or sometimes mice. But the idea is that you've done a genetic screen for a specific phenotype and you've isolated a large number of mutants with the same phenotype. And the question that you're trying to ask is the same gene mutated in all of these different isolated mutants? And it can be used to, one, predict the total number of genes that influence a trait, and it is used to define a complementation group where you have multiple mutants that are affecting the same gene. So as always, I feel like it's best to understand this stuff with a real life example. And so an example of this is in Drosophila, so these fruit flies. And so, normally Drosophila fruit flies have wings, and you are a geneticist, so imagine yourself as a geneticist that's interested in understanding how wings develop. So how does the fly know where to put these wings, what is used in order to build these wings? And so you do a screen where you mutate flies, mutate wild-type flies, and you isolate flies that don't have wings. And so here are an example where you have isolated two different mutants. So these are completely unrelated animals, and they have the same phenotype. And the same phenotype is the fact that they don't have wings. Now you want to know, do these flies lack wings because they have mutations in the same gene, or are different genes affected? And so we are distinguishing these two cases. So in the first case, the mutants are in different genes, and in the second case, the mutants are in the same gene. So here we have gene 1 and gene 2, and so we can see that this fly has a mutant. It's homozygous right here in the beginning of gene 1, and then this one is wild type. This one has a mutation in gene 2, and so again, it's homozygote. And that is at the end. In the second case, the location of the mutation in the first fly is the same, but now the mutation for the second fly is also in gene 1. So it's in a different location, and so this actually would define different alleles. So the MA allele is different from the MB allele, but it'd be two alleles in the same gene. So how do you actually determine whether it's the first case or the second case? And so what you do is you actually cross these two genes. Sorry, not cross these two genes. You cross these two mutants together. And so in the first case, you would have an F1 with the following genotype. And in the second case, you would have a genotype, an F1 with the following genotype. So one of the assumptions is, for complementation, is the wild type allele is dominant, okay? So this interpretation requires knowing that the wild type allele is dominant. So in this case, the genotype is this. And in this case, we have a wild type copy of gene 1, and we have a wild type copy of gene 2. And so this is called complementation because when you cross these two mutants together, they complement each other because each one is able to provide with a functional copy of the two genes. And so these wings develop just as normal. In the second case, complementation doesn't occur. So what happens is you end up having two good copies of the second gene, but in the case for the first gene, neither one of these copies are functional. So neither one of these alleles is functional. And so these F1 flies do not have a functional copy of the protein 1 that is produced by gene 1. And so in this case, no complementation occurs. And so you can recognize that and because the flies are mutated. And so the idea behind this is that you would extend this and you would isolate something like 30 different mutants, and then you would define complementation groups. So you would have complementation group 1, and you would have 8 mutants that belong to that. And that would mean that all 8 of these mutants are mutated in the same gene. And then the same thing with this. And so by doing this, you could say, okay, there are 4 genes that we can isolate that we know have an effect on wing development. I've isolated 8 of these that belong to this gene, 6 that belong to this gene, 7 that belong to this gene, and 9 mutants that belong to this gene. And so as a researcher, then you could have 4 grad students each follow up on one of these individual genes as research projects. So that's the idea behind complementation analysis, how you perform it, and kind of the logic behind why you would do it. All right, the final case that I want to talk about is what if a gene is on the X chromosome? And so normally we have been assuming that animals are diploid, and when you have a mutation, this individual can produce 2 different types of gametes. And so you do like a Punnett square that kind of looks like this. Okay. So what happens though if the gene is on the X chromosome? Because males only have 1 copy of the X chromosome. And so when they create their gametes, they pass on 1 gamete is the X chromosome and 1 is the Y chromosome. And so this actually skews how this is inherited. And so what this means is that the way it's inherited is dependent upon the sex of the offspring. So you actually have to phenotype the male and the female separately because of that. So let's talk about an example of that. So what if the gene is on the X chromosome and it controls the eye color? And so let's say we have a pure population and it looks something like this. So typically we would draw something like this. So there's a gene on the X chromosome. There's a wild type copy of it, which we're just going to call X. And these flies are red. And then the males have this white allele on their X chromosome, and then they have a Y as their kind of their complementary chromosome. So the Y chromosome does not encode this gene. So for this gene, the males are effectively haploid. So we cross these together, and so all of the individuals are red as a result of this. We could also do the opposite cross, where we cross a female white fly to a red-eyed male. And so in this case, the genotype would look like this. So the white allele is recessive, so the female fly must be homozygote, and then the male has red eyes, so it must have the wild type copy on its X. So in this case, when you do the cross, you have half of the offspring are red, but they are all female. Half of the offspring are white eyes, but they are all male. And so one of the things you'll notice about this, if it's on the X chromosome, the direction of the cross matters. Does it matter what's for the sex? If it's a male individual crossed to a female, you have to make sure you know which phenotype is which. And so just by this simple demonstration. So how does this work? So this individual has to produce this. This individual has to produce either this or this. So the female flies look like this, and then the male flies look like this. So the female flies are red because they're heterozygote, and then the male flies have red eyes because they have the single copy of the wild type gene. So in this case of the cross, this allele has to be passed on, and then the male can either produce this or this. So in this case, we produce this. So these individuals are female if they receive the X chromosome. And then the genotype of these individuals would be this, so they're male, and then they have wide eyes because they don't have the second version to complement that. And then you can continue to do this cross into the next generation. So if you intercross this and this, then you can also go on and do a Punnett square to figure out that you would get these following ratios. Sex-linked traits can also be there are for human diseases, there is also a large number of examples of sex-linked traits. So hemophilia and color blindness are two kind of classic examples of that. And in the activity, we'll kind of cover applying sex-linked traits to pedigrees. But one of the important things that you need to do is use this information right here in order to try to distinguish between situations where the gene is autosomal or sex-linked. So this, again, is an example of a sex-linked trait, color blindness. So if you're colorblind, you would not see the number 15 within here. And this is an example of a X-linked trait. So this is recessive, but you'll notice that you have a situation where you can pass on the disease. I'm sorry, that's not correct. So you'll notice, for example, you'll have situations like here where all of the male children of a female that has it have to have the trait. So for example, if this was not sex-linked, then if you had this cross right here, you could have individuals that, you could have male individuals that did not have the trait. All right, so this is chapter 4, and that covers the non-Mendelian inheritance part of the class.